There we go. Okay, so hello everyone. Thank you very much for being here. So, and thanks for the introduction, which allows me to go straight to the presentation. So the introduction is as follows. So we are interested in dynamics of extracellular metabolites because their concentrations over time, because they provide a footprint of the physiological activity that takes place into cells. In particular, what is of our interest is to look at the growth, uptake, and excretion rates of extracellular metabolites, because they can be related with intracellular metabolic fluxes. And however, the point here is that in experiments, what you can rather easily observe is the extracellular metabolite concentrations, whereas the corresponding uptake and excretion rates are not so obvious to actually quantify, because the data is typically noisy and sampled, and there is non-trivial correlation among the dynamics of the evolution of concentration of different metabolites and the growth rate. And there are typically discontinuities in the profiles of concentration of different metabolites that relate with actually with sudden changes in the physiological and metabolical activity of cells, which you do not observe directly because you don't have access to the internal dynamics of the cell. So the problem here is to, given data of metabolite concentration evolution in a culture and given growth measurements of a population of cells, essentially reconstruct uptake and excretion rates as well as growth rate. So what is available today? How are these things done? Where typically most methods look at rates in balanced growth conditions, where essentially the rates are constant. And those methods who actually look at dynamic growth conditions, where growth rates may change over time due to changes of metabolic activities, are affected by a number of issues typically, not all of them, all of the issues of course. Typically what people do is to take noisy concentration profiles and in the basic case perform some sort of non-parametric fit and then numerical differentiation, or otherwise fit a priori determined fixed parametric curves, which introduces some sort of like a priori unjustified, not necessarily fully justified hypothesis. Then another typical assumption is to perform separate processing of different metabolic profiles, which kind of like ignores the inherent correlations among them. And then yet perhaps the most critical point is the detection of switches in metabolic regimes, which are not, which basically change the dynamics of metabolite exchange between cell and medium. So the point of this work is to try to come up with a sound and comprehensive framework to address this dynamical estimation problem by addressing these different points that or limitations of current approaches or traditional approaches that I'm mentioning here. And the way we propose to do it is via Bayesian estimation techniques performing regularized estimation. And let's see how. So given this context, now this we go on as follows. We look at the approach for dynamical modeling of growth and metabolite uptake and excretion. Then we look on the basis of this method, of this dynamic modeling approach, we discuss our estimation method, and we look how that works on validation data, and then we look at actual results on experimental data, and to the extent it is possible, how to validate these results given that the true rates are actually not known. Okay, so first step is to model the system of our interest, the system meaning the dynamics of metabolite exchange between cells and growth environment. So we look at a fairly basic model, if you wish. It's you see it here in the red block. It is basically one equation describing growth rate mu that essentially determines how the biomass B changes over time. And then we consider possibly n metabolites being exchanged between the environment and cells and a cell population, and this exchange is basically described by n different equations in the state variable Ci. So Ci is the concentration of a metabolite I into the medium that is exchanged at the rate R times the actual biomass present in the growth medium. So the more cells, the more exchange, proportional to rate I. So our unknowns basically will be the growth rate mu and the exchange rates Ri for I that goes from 1 through n metabolites. Based on measurements, so what measurements do we consider? Well, you have here an example in the bottom right plot. Here you have, for an example with this is simulated data. For an example with 2 metabolites, you have a typical growth curve in the first plot here. So you see the biomass amount showing exponential growth until a first arrest. Essentially here you see in the second, it's a primary carbon source that is being consumed. And when this goes to 0, you have sort of a growth near arrest. A second metabolite is then uptaken. And then when this also is exhausted in the medium, you have complete growth arrest up here. So the type of data we consider are sampled and noisy measurements of biomass and all the metabolites you have over time. So this is a dynamical experiment like here. Yes, we consider not necessarily the measurement times have to be the same for the different metabolites. We consider that there is a measurement error on top of these measurements, which we consider to be Gaussian for simplicity, but not crucially. So we go now to the problem of what to tackle. So this previous model here can be comprising n+1 equations, can be summarized into a sort of a state space model of this kind, where X is the full state including biomass and the n concentrations at time t, and the unknown rates, which are the growth rate and the exchange rates Ri, are collected into an input vector u, which is basically the thing that we want to estimate over time. So this can be summarized into a state space model of this kind. Dynamics are in the first equation, and the measurements we get is basically at any time where we get a measurement, tie in measurement set tau, you get a readout of this state plus noise, okay? So the problem here is to find u over time, its evolution over time, that best explains, in some sense, our observations y. This is a loose statement of the problem, because the question now is, okay, but what are the possible uses? So the problem itself is not well specified, because you have like a continuous multidimensional unknown with sample and noisy measurements. So there is no unique solution that fits the data in principle. So what are the possible uses before we run into nonsensical estimations? How do we make sure that the u we pull out of this problem is not essentially explaining noise because it performs overfitting of the data we have? And how do we cope with the fact that u is something that may be smooth over constant metabolic regimes, and then it may change abruptly at times where actually metabolic regimes change in the cell physiology? So we need some sort of, to address this problem, we need some sort of like suitable characterization of the possible use that we are actually seeking. So we take, as we said, a Gaussian process approach, which is like a specific class of Bayesian approaches. So we assume here, so we give a statistical characterization of u, so the unknown rate profiles, which is purely instrumental to set up the problem. So it is a stochastic characterization, which doesn't mean that u is a stochastic process in itself. It's just a tool to introduce a priori information that regularizes our estimation problem, okay? So in particular, we assume that our unknown, so each of the entries of the unknown state u, ui, is a Gaussian process. What Gaussian process? Well, it is assumed to be, as you see, a double integral of a white noise process, okay? Modulated by a parameter that may also change over time t. This parameter is a tuning parameter for us. We'll come back to this into detail, whose purpose is essentially to determine how variable this rate can be at any point in time. Okay? So you can see that gamma plays a role of like a multiplicative factor over this noise, so this stochastic characterization of our unknown input. So the larger the gamma, the larger the possible variabilities. Okay? And this depends on time. So to give you an example here, so this technique allows you to basically introduce some information on what you are seeking. In particular, you know that because this is a double integral of a white noise formally, this is a non-parametric form. I'm not saying that a specific trajectory will have to be a function with such and such parameters. It's a purely stochastic description, which, however, will in any case lead to continuous profiles of U, and we may actually tweak the expected regularity of this unknown input. How? Well, here I have a little plot. You see that we have a probability scale here from less probable profiles that we are seeking and most probable profiles. Here you have this over time. You have this gray line that is our tunable factor gamma here. And I'm saying that basically at these times we may expect bigger variability of these unknown inputs. Correspondingly, what you are saying a priori with this characterization is that U cannot fluctuate arbitrarily over any point in time. So this light green curve here has very little probability a priori, whereas it is allowed to go sudden changes around the times where this gamma is said to be high, which means that these two curves here are both of a large probability a priori, okay? And now we'll see, of course, how do we instruct the algorithm so that our estimation, how do we basically choose this gamma so that it makes sense for the problem at hand. So, okay, so here's the characterization of the unknown input rates. It's a statistical characterization. And you see that basically it's a dynamical characterization. This is like a second order linear system. So if you want to collect everything into a state space system, you can write up a system of this form. Again, you have like a first dynamical equation that basically encodes the dynamics of these unknown rates U. And then U is simply a selection of these state entries. If you connect this characterization of your unknown inputs with the previous model we had, which I had here for the biomass and growth rate that you observe in your experimental medium, what you end up with is a bigger augmented dynamical system, which has that depends on this regularization profile gamma, and it has measurements that are the actual measurements you collect in your experiment. Now this system is a stochastic differential system where the state contains the unknowns. Therefore, our estimation problem, the problem of estimating the unknown rates over time, becomes a Bayesian estimation problem. So, which formally reads as, to get your best estimate of your unknown rates, which are in this state Z here, you have to compute the conditional expectation given the data and your choice of the regularization profile here. And this can be done actually by a Kalman smoothing approach, because everything is expressed in terms of a dynamical system. And it's a Kalman smoothing. It has a forward iteration, which is standard Kalman filtering, and backward iteration that actually corrects the data so as to make sure that you are using your data at full at every point in time where you want to estimate the unknown rates. So one important point is, how do you choose the regularization profile so as to instruct your filtering that your unknown states may undergo abrupt changes at times? Well, it's a data-driven procedure. You take your data. In a first step, by statistical hypothesis testing, you check where metabolites actually undergo, for instance, they drop to zero, where it's typically a condition where metabolism changes. You fix these time windows where things drop to zero somewhere. Then separately, you take the same data, you operate a first fitting with standard splining techniques, and take derivatives to get a first handle on what magnitude the rates you're looking for may have. This gives you these gray lines, which are basically an overall smoothness that you expect for your unknown profiles, except at the switching times that you have detected before. At the switching times, you take your regularization profile and you push it up in these intervals where things change abruptly, so that there you allow estimates to change abruptly. Okay. Now, if we look at simulation results, we're here from this for the same data I showed you before. This data, you see that with this approach, what you get here on the left are actually the estimates of the growth rate in the first box and the two metabolite exchange rates in the second two subplots. And you see that we are capable of producing very smooth estimates in regimes where metabolism does not change abruptly, as well as to reproduce abrupt changes at the times where things actually do change because of metabolite dropout in the environment, for instance. Along with credibility intervals, which are like essentially these confidence intervals that were mentioned in the first presentation. And on the right, you see actually these estimates compare with the true rates that we generated in simulation, which are the dashed black lines, which are essentially in perfect agreement, and also the estimates in blue that we used for the tuning of the filter, which is also the typical approach by which people do this type of estimation, which are actually not in such of a good agreement. But for our filter, this is sufficient to instruct the Bayesian prior sufficiently well to actually let the data speak eventually. So when applied to actual experiments, so on the left you have a dioxy shift experiment in E. coli. So E. coli is growing on glucose, and at depletion of glucose, the acetate that had been excreted up to that point is uptaken again with some residual growth. And you see that the estimates, okay, they have the same qualitative shape that we had before, because the simulation was actually a simulation of this experiment, and they make qualitative sense. Quantitatively, how can we check that these that these rate estimates make sense? Well, we actually took, based on these rate estimates, we took the values estimated before the sudden switch of metabolic regime. We used those values to make a metabolic flux analysis and compare it with the equivalent analysis made on the basis of the smoothing of the signals operated by splines. And essentially what we see in these bars is that the uncertainty in internal flux estimates that we get from our estimate of the external rates are much better constrained in the case of our algorithm, which is basically reflected in these very narrow red lines, bars at the bottom, compared to the corresponding analysis operated on standard splining procedures, the gray boxes. So with this I am done. I just want to conclude briefly that this is like essentially a fully automated procedure, whose software is actually available, and we have further experimental evidence that this procedure makes sense. There is room for robustification because extended Kalman filter is per se not the most robust algorithm in the world, but this is just one, let's say, one piece of our overall framework that can be replaced and improved. And also that this approach applies to much more general dynamical models. Thank you very much for your attention.
Questions?
I was wondering if the parameter U does it really have to be a function of time in your model, or can you model that as a constant but that is modulated by another component, say part of the biology that's over time modulating and then that process could be parametrized so that
it is also possible. So this gamma here, which is essentially what determines the regularity profile of our unknown rates. I mean, if it was constant, you would not be able to capture smooth, abrupt changes. And this is actually what traditional Gaussian process approaches do. This parameter is constant. The key here is to have it time varying. How do you set it? We set it by a data-driven procedure. If you have more biological information so as to instruct it better based on other experimental relevance, the better you can use it.
About the optimization, so there are regularization-based approaches that are used, for example, in image processing, where you incorporate smoothness constraints as a penalty term
Yes.
in your optimization.
Mm-hmm.
Have you tried something like that here?
One thing that I haven't had the possibility to include here is the fact that parameterizing an unknown input as double integral of white noise is essentially equivalent to a Tikhonov regularization approach where you have a second order penalty term, and the penalty factor becomes this gamma factor here. But this setup allows me to tackle the problem by the dynamic nature of the resulting augmented system, and so I can apply Kalman filtering and everything becomes dynamical.
Thank you very much for your presentation. I had also two questions kind of going in a similar direction. The first one, actually, I was wondering, I mean, you use the data essentially twice. So first to construct your gamma, and second to do the estimation. So I was wondering about statistical independence and overtraining models. I mean, you show that it nicely works. I don't know whether you consider that. And the second thing is about scalability.
Okay. About overfitting, well, in the first step, which is the tuning of the filter, we are basically building a Bayesian prior, which is, and I haven't said that how weak this prior is. We are using a very weak prior built on top of this procedure. So step one is really a weak instruction of the filter. So there is no perfect independence, but it's basically two different things. The second question was scalability. Scalability scales essentially linearly in n. The biggest concern is the stability of the extended Kalman filter. If you want to make things more robust, you have to use, let's say, unscented versions of the filter that scale still linearly with a worse coefficient, let's say.
Thank you. And now we are going to move to the last